and see who's showing up. Um, okay, let's get started. Thank you everyone for being here and good afternoon. Welcome to the penultimate Biome seminar for the semester. If you are joining us for the first time, Biomes is YSE's weekly research forum, bringing together students, faculty, visitors, and staff to share ideas, engage with cutting-edge work across environmental science, policy, and practice. Today's talk is going to be about how forests respond to multiple and interacting disturbances and why understanding those interactions is really important for predicting ecosystem change and informing environmental management. Our speaker today is Dr. Charlie Driscoll, a university professor of environmental systems and distinguished professor in the Department of Civil and Environmental Engineering at Syracuse University. Charlie is an environmental engineer and ecosystem scientist whose work focuses on the biogeochemistry of forested and aquatic systems, with a particular emphasis on how air pollution, climate change, and other disturbances may affect ecosystem structure and function. Over the past 4 decades, Charlie has been a leading figure in understanding the impacts of acid deposition and mercury pollution on forest and freshwater ecosystems, in linking long-term environmental change to both ecosystem process and environmental policy. And his work is really deeply rooted in long-term and place-based study, particular in his work at the Hubbard Brook Experimental Forest, what we'll hear about more today, and one of the most influential sites in ecosystem science. Hubbard Brook also has a very strong connection to this school. So those of you who don't know, it was established in part by former YSE professor Herb Borman, who helped to pioneer the watershed approach for studying ecosystems and helped co-found Hubbard Brook in the 1960s. Other Yale faculty like Tom Sycamore have been involved along the way. And so it's really meaningful to be able to bring Charlie back here and connect us with the work done at that site and the legacy that we have here at the school. And so without further ado, please help me welcome Charlie Driscoll.
Well, good afternoon, good evening. Can you hear me okay? Okay, good. Well, what's that? Can't hear? Oh, okay. Okay, yes, you're right. I think you told me. How's that? All right, very good. So thank you, Robbie, for a nice introduction. So when Robbie approached me about giving a talk, I gave him a list of possible topics, and he goes, "What are you, crazy? You have to talk about Hubbard Brook." So that's what you've got. If you don't like it, you have to blame him. But I'm happy to talk about Hubbard Brook. I haven't been at Yale for a while, so this is a great opportunity to come back and talk a little bit about Hubbard Brook. It's also a great opportunity for me because Hubbard Brook is a LTER, long-term ecological research site, and a lot's going on. And so I'm a little worried about this talk. It's fairly complicated, but you're smart people, so but I look forward to getting some feedback on the talk. So this is what I'm going to try to cover. I'm going to give you a little bit of an outline on the history of Hubbard Brook, and then talk about the research approaches. And historically, we've had sort of 3 independent research themes looking at the effects of disturbance on the structure and function of the forested ecosystem: climate, air pollution, and biotic disturbance. But what I'm trying to do here, which is sort of a work in progress, is to integrate across these disturbance regimes and try to talk about what are the primary drivers, what are the effects, what are the interactions, which makes it fairly complicated. So I'm going to throw a lot at you. So I'm just giving you warning. So Hubbard Brook is established by the U.S. Forest Service. It's in the White Mountains of New Hampshire. It's about 3,000 hectare. It was established in the mid-50s by the Forest Service, principally Bob Pierce, with the motivation that empirical evidence in New England in the 50s was that forest cutting would either cause flooding or cause drought conditions. And so they want to understand what would be the hydrologic response to forest cutting as it would influence water supply for southern New England. So that was the basis of the study that got started. And then Bob Pierce was interested in expanding the scope of the study. So in the early 60s, he ventured out to Dartmouth and talked to Herb Borman and Gene Likens, who are faculty at Dartmouth at the time, and encouraged them and their students to work at Hubbard Brook. And they had a few fits and jerks at the start, but they eventually got some NSF funding in the early 60s to start the measurements through the small watershed approach that we'll talk about in a few minutes. This study was one of the first reports of acid rain in North America and prompted a lot of research on acid rain effects. And then more recently, but still a long time ago, in the early 80s, Tim Fahey and I put in a proposal as Hubbard Brook being a long-term ecological research site, and it was funded, and it's continued today. So I wanted to have a shout out to Yale, as long as I'm here in Yale, to talk a little bit about the history. These are some photos. This very old photo up here shows the three principals. So this is Herb right here. This is another photo of Herb, and this is Gene Likens, and this is Bob Pierce in the middle. And then I've got my former colleague, Tom Sikkim, who used to be a dear friend of mine, spent a lot of time with him. And a lot of the vegetation work that goes on today was really set up by Herb and Tom and students at Yale. So there's this long history, and we're hoping we can get even more scientists from Yale working at Hubbard Brook going forward. So hopefully this will prompt some interest in some of the topics that we're working on. So this is a map of the forest. It's a sort of a bowl feature area. As I said, it's about 3,000 hectare. There are a series of experimental watersheds that are shown here. This is a slide taken a number of years ago looking from the base of the watershed out into the experimental watersheds. You can see if you've got a good eye up here, this is one of the cup watersheds in here. This is watershed 4, but this gives you an idea of the landscape. The area is underlain by granitic bedrock. It was glaciated, so the soils developed under glacial tills. They're acidic soils, spotosols, very low base saturation, very acidic soils. The forest was cut about 100 years ago. It's a northern hardwood forest and has a humid continental climate. This is a map of what the forest looks like. You can see in the red coloring, that's mostly northern hardwood forest. And then there are conifers at higher elevations here, mostly balsam fir and red spruce. Then some hemlock down in here at the base of the watershed. This is just a few photos of the forest, just to give you an idea of what the forest looks like. But I put out a couple photos here and here so that you realize that the beech are doing very poorly here through beech scale, and we're anticipating more problems with beech through beech leaf disease, which is anticipated to impact the forest in the not too distant future. So the area of the northern forest is shown here in dark green. So it really covers areas in the Adirondacks, an area that I also work a lot in, but also in northern New England and also in the upper Midwest. So some of the early work, particularly work that was pioneered by Herb and Tom, had to do with characterizing the forest. They worked with colleague Bob Whitaker, who was at Cornell, to try to characterize the forest, the species, and the stoichiometry of the forest. So this is a time series of the above ground biomass and the distribution of major tree species that was started by these early investigators, but continues today. And you can see early on the forest was a strongly grading forest, and then it sort of abruptly stopped. There were some damages and events that occurred in the late 1990s, there was a severe ice storm that had a huge impact on the forest, and a microburst about 10 years later that also had a huge impact on the forest. But essentially, the above ground biomass is essentially in a constant condition. But there's been large changes in the species distribution. So early on, it's largely three principal hardwood species, yellow birch, sugar maple, and beech, but they've shifted substantially over time. Early in the study, sugar maple was very prominent, and now it's tanking, doing very, very poorly, and it's being replaced by beech. This is in the reference watershed. As I mentioned, the soils are acidic spodosols, so they have a thick organic horizon, very, very low base saturation. Um, there's a lot of spatial variability, even though the watersheds are relatively small, on the order of tens of hectares. We see a lot of variation due to effects on the hill slope that result in lateral flow and also in the near stream channel, which influences soil development. So this is a map showing you the spatial variability of the soils. So along the ridge caps, there's a lot of lateral movement, all in here shown in the browns and gray colors. And then the typical spodosols are shown in yellow, sort of in the middle here. And then there's a lot of also development along the stream channels. Some of these are ephemeral stream channels where there's a lot of high accumulations of organic matter. So a lot of spatial variability. Sort of the instrumental and experimental focus has been on the small watershed approach. So then when Bob Pierce set up these watersheds, he set them up as experimental units. So the idea is that there's a gauging station, which is anchored to the bedrock, and most of the flow leads through the stream flow, and then a series of precipitation stations throughout the watershed. So to try to do a detailed quantitative water budget of measuring precipitation coming in and stream flow going out, and then the difference being evapotranspiration. The initial contribution from Borman and Likens was to measure the chemistry associated with precipitation in stream water, and then use this sort of hydrologic framework to do watershed mass balances. And then to try to use these as experimental units and do various experiments, relatively large scale experiments. So this is a slide giving you the topography of the reference watershed, watershed 6. And so just to orient you, the gauging station is down in here, and then you go up to the headwaters at the edge of the watershed. So you can see the deposits of till, relatively thick in lower elevations, and it grades out to be very shallow up at the top of the watershed, and then you can see the canopy height. And we usually divide these units into zones. So the lower elevations are the hardwood zone, and then there's a transition, and then conifers at the highest elevations. So Likens and Borman put in these sort of low-tech precip collectors for precipitation chemistry to measure bulk deposition, and we still use something similar to this today, and we swap these out weekly. So these samples were started in the 1960s, and they continue on today. And then a series of gauging stations. So this is a modified San Dimas flume. You can see a V-notch weir here, and then there's a flume back in here to measure continuous flow. So if you look at the streams, people have heard of Hubbard Brook, but you go there, you see the streams, they're actually kind of dinky, maybe not too impressive. This is what they look like. And then we have these patterns, these temporal patterns. And for years, I always thought Hubbard Brook was very resilient, never really changed, not very exciting. But in recent years, we've seen some rather abrupt changes. But historically, the water budget over the annual cycle looks something like this. The precipitation, which is shown in triangles, was relatively flat throughout the year, pretty much uniform precipitation throughout the year. The hydrology very different. So we have the highest flows typically early in the study during snowmelt. So peak flows during April, decreased flows during the summer because of losses from evapotranspiration, and then would increase in the fall, and then lower flows in the winter because of snowpack accumulation. So the approach that we use for experimental study and to answer questions is a fairly basic approach. It's a multi-tiered approach. We do long-term measurements. So we go in, out. I'm going to show you a lot of those types of observations. We do experiments using primarily watersheds as units, but we do small scale units. We also do modeling. We do cross-site studies. I'm not going to talk at all about modeling today, but I'm going to talk about the others. This is a listing of our experimental watersheds. We have 10 experimental watersheds. They've been established at different times. We have two reference watersheds. I could give you long stories about why we have two, but I'm just going to tell you we have two. We have a biogeochemical reference watershed, watershed 6, and we have a hydrologic reference watershed, which is watershed 3. Then a number of the watersheds have been experimentally manipulated. The first experiment was done on watershed 2 right here. And it was a little bit of a weird experiment. And the idea from Borman and Likens, they wanted to look at the role of vegetation in controlling the hydrology and stream water chemistry. So the idea is they would cut the forest, leave the biomass in place, and then apply a herbicide for 3 years to prevent regrowth, and then allow it to do what it's doing. So that was the first experiment. The next experiment was a strip cut here in watershed 4. And that was done a more benign treatment where there were 20 meter strips that are parallel to the elevation, a third in 1970 cut, a third in 1972, and a third in 1974, and a buffer strip along the stream channel. Another cut experiment was watershed 5 here. That was a very severe treatment, a whole tree harvest that I'll talk a little bit about as a commercial operation. And then there was a block clear cut in 1970 as well. Then the last experiment, which was not a cutting experiment that I was heavily involved in, actually still involved. And we have some people from Yale working on this a little bit as well, which I'm happy to see. And that is in watershed 1 that we did a calcium silicate addition in 1999. And those experiments, even those were done over those times, they continue on. So one of the advantages and disadvantages of Hubbard Brook is experiments never end. We just keep measuring, 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 for good or for bad. So this is a shot of the valley looking down sort of from the north facing over to the south facing watersheds. This is watershed 2. This is where the cut and herbicide treatment was done. This is watershed 4. If you've got a good eye, you can sort of see the strips here and you can see the buffer strip. And this is watershed 5, the whole tree harvest. So this photo was taken around the mid-1980s, shortly after the whole tree harvest. The whole tree harvest was a very severe treatment. You can see the skidder trails along there. And then if you've got a good eye, you can see these sort of blotches on the landscape. Those are various meteorological stations where we measure precipitation and temperature and things like that. Okay, just a few photos of this. So this fellow right here, Tony Federer, he is applying Agent Orange to the watershed 2 treatment. You notice no mask, no gloves, nothing, and he is alive and well today, doing his thing, very robust character. So anyway, and this is a photo of the whole tree harvest to give you an idea of the scale of operation. The trees are cut off at the base, collected, drawn down at the base of the watershed, and then shipped up and exported. A very large, comprehensive removal of the above ground biomass. And so we've tracked these things over time. This is sort of an integration. This is similar to what I showed you before of the above ground biomass for the uncut sites, but then we have these cut sites and we are tracking their accumulation over time. And we've looked at overall biomass, but we're also looking at species distribution. Now, I know this is a very horrible slide to show you. This is the changes in species, tree species, after the cut in watershed 5. So they happened in the early mid 1980s. The initial tree species were mostly beech and maple. And then you can see, well, maybe you can't, but there was a large increase in pin cherry. Those have died out. And today, beech in particular has come back. Sugar maple, as we'll talk about later on, is not doing very well. So we track species distribution, not only their growth, but also their survival over time. And I'll show you some more of that. A little bit about my favorite experiment, the last night experiment. We argued, that's one thing about the Hubbard book. We have about, give you an idea of the study, there's about maybe 20 institutions, maybe 70 people who work up there. They feel very passionate about these experiments, and we have to argue about how we do them. So I told you at least 2 years arguing about what we were going to add for this. We talked about adding limestone. Steve Hamburg wanted to add calcium sulfate. And eventually we decided to add calcium silicate, which I was fine with at the time. In retrospect, turned out to be a very fortunate decision because of the interest now in using silicates for carbon sequestration. But at that time, we were interested in adding back calcium that had been leached historically from the forest from acid rain. So historically, we thought that the base saturation of the forest was about 20% in the mineral soil before the advent of acid rain, and today it's about 10%. And so we wanted to put back the exchangeable calcium that had been lost from historical leaching. So we went around the world. We got samples from different wollastonite mines to look at the isotopic characteristics that we could use as a tracer. The best mine was a mom and pop operation in Governor, New York, which was barely in business, but this is a photo of that. We got very fine grained material. We had it shipped to Illinois to have it pelletized, and then we applied it to the site. So the idea is we wanted to use a tracer to try to separate out the natural weathering from the wollastonite weathering. And as I mentioned, to try to experimentally put the base saturation from 10% up to about 20%. And that required application of about 30 tons. Being a good engineer, Robbie told you that I'm an engineer. We always add a safety factor, so 50% safety factor. So about 45 tons or about 4 tons per hectare. So the operation looked like this. We had about a bunch of these 1-ton sacks. It was applied by helicopter, and this is what the application looked like. And we've continued to study that, and I'll show you some results of that a little bit later in the talk. So getting back to the Hubbard Brook ecosystem study. So I mentioned that Hubbard Brook is one of these LTER sites. This is the conceptual model that we have for the study. We have 3 drivers of change that we've looked at sort of separately: climate change, air pollution, and change biota. And we're interested in how these disturbance regimes influence what we call the biogeophysical template, which is sort of a fancy name for the landscape. So this includes the topography, the soils, the vegetation, the historical disturbance regimes. And so if you look carefully at this, you can see that this sort of has a temporal element. So the template is, we are envisioning, is changing over time in response to these disturbance regimes. And then we're particularly interested in the functional responses. How does the hydrology change? How do the vegetation patterns and how it functions change? How do the food webs change? How does the biogeochemistry change? And for biotic change, there are feedbacks here through these changes. It changes biota and it changes the landscape. So this is our theme. But I'm learning, particularly in recent years, that we can't really separate these. They're all going on in this landscape, and they're all interconnected. So I really am interested And that's sort of the theme of this talk. What are the interactions and what is the interplay between these different disturbance regimes, which makes for a very complicated study. But just a little bit of background information and show you some of the data sets. So first, under climate, Hubbard Brook is a relatively cool place. It's a relatively wet place, has a significant snowpack. We have, however, seen a lot of changes, as you might expect, and these changes are very similar to what we would expect for northern New England: increases in temperature, increases in precipitation. In terms of and how this is occurring, we're seeing the most clear evidence of increases in temperature during the winter time. The trends are clearest during the winter, less clear, particularly during the fall, but we see increases in temperature in every season, but particularly in the winter time. Precipitation is a little bit different animal. As I said at the beginning of the talk, historically, precipitation was uniform throughout the year. That has completely changed now, where we're seeing increases in precipitation, but those occur virtually entirely during the summer months. So completely different. Because it's getting warmer, particularly in the winter, we're losing snowpack. So down any measure of snowpack, the quantity, the water content, the temporal distribution, they're all decreasing. A lot of year-to-year variability, as you see, but because we've been at it for 50 years, we can determine statistically significant trends. Because of this change in climate and snowpack, we're seeing changes in hydrology. We're seeing the flows are getting higher. The low flows are getting higher. The high flow regimes are getting higher. So there's it increases. And so the changes in the distribution of discharge have completely changed. So less and less very low flow conditions, more and more high flow conditions. We're seeing peak increases in peak flow over time. So the peaks are getting higher, and the nature of these high flow conditions is changing. So when I first started in the 70s, the highest flows always occurred during snow melt or on rain on snow events. Now, this is the 20 highest flows in the history of Hubbard Brook. All sorts of different hydrologic events. So they could come in the summer, they could come in the fall. Throughout the year, we're getting high flow events. So the character of the hydrology has completely changed. So this is a long-term plot showing you the annual water balance. So red is the annual precipitation, blue is the stream water loss, and the difference between the two in green is evapotranspiration. So you can see a lot of year-to-year variability, but the precipitation is marching up over time, and the stream flow is responding to that. So for decades, the difference between uh, precipitation and stream flow were remarkably constant, and you can see that in the green. But in recent years, that has completely changed. So you can see that a little bit. This is for one watershed. This is, I don't quite have this updated, but this is a 3-year running average of ET, just ET, the difference between precipitation and stream water chemistry for the 4 reference watersheds. So 2 uh, south-facing, 2 north-facing watersheds. So you can see starting around 2010, this marked increase in ET, which has happened and persists today. We're seeing changes in the phenology. So we're seeing leaf out in the spring getting later and later and later, and leaf fall, leaf senescence getting later into autumn. So we're seeing about a 3.5 day per decade increase in the growing season. Again, a lot of year-to-year variability, but statistically significant changes. So we've seen these chronic changes that I've talked about, but we've also seen episodic events which are important, such as the ice storm in 1998 that I mentioned and the microburst in 2013. So a little bit on biogeochemistry, which is sort of my passion. We're seeing all sorts of changes. So this is increasing CO2. We know about this. These are data from Harvard Forest. We're also seeing some interesting observations. These data are soil respiration. These are Peter Groffman's data. And he started working at Hubbard Brook in the 90s. He has a number of microbial soil assays. These are soil respiration. And for years, relatively constant, some year-to-year variability. But then starting in 2015, we've seen this striking increase in soil respiration that we're trying to figure out what is going on. As I mentioned, air pollution is one of the research themes at Hubbard Brook. We've seen a lot of changes over time. This is a map showing you data from the US National Emission Inventory, showing you emissions of sulfur dioxide in red, nitrogen oxides in blue, and ammonia in green. And so you can see since the 1970s that there have been huge drops in sulfur dioxide. Nitrogen oxide's relatively constant until about 2000, and then it's been dropping like a rock. And ammonia, and I have to say, these emissions for ammonia are horrible. They're not very good at all, and they're probably gross underestimates, but I just include them for sake of completeness. So these are the precipitation data for sulfate, nitrate, and ammonia at Hubbard Brook, annual volume weighted concentrations. And there are pretty good relationships between national sulfur dioxide emissions and sulfate at Hubbard Brook and national nitrate emissions and nitrate at Hubbard Brook. Although it does look like in recent years, the values are leveling off. So maybe we're reaching a low value. In fact, the values of sulfate are very similar, maybe even below what we think are background values. These are long-term changes in stream water chemistry in the reference watershed. So the upper panel is sulfate. You can see that's dropping, consistent with decreases in atmospheric deposition. Nitrate, which is pretty wacky. It was high, increased in the 60s, high in the 70s, and then it's dropped down to very, very low levels. There are some blips here. Like I mentioned, this we think is a soil freezing event. This was the ice storm. We're not sure what this is. It may be the microburst, but you can see that most of them are pretty low. And these decreases actually preceded decreases in atmospheric deposition. Remember, I said that those really started around 2000. So these decreases were before that. We've seen large decreases in calcium and other base cations. I'll talk about that in a second. And then the last panel is alkalinity, which is the acid-base chemistry. So it's still acidic, negative values of alkalinity or acid-base of alkalinity, but it is increasing because of decreases in acid rain. We've seen interesting changes maybe in response to this. So these are some of Peter Groffman's data. These are showing two measures of mineralization dating back to measurements that were made by Herb back in the 70s. So we see decreases in mineralization. We see decreases in nitrification over time. And we are thinking about what's driving those patterns. So that's in the back of my mind. Why is the nitrogen cycle changing? So we hypothesize that this is partially driven by increases in carbon inputs to the system. If you remember, I told you previously that carbon dioxide concentrations are elevated, so there's probably a carbon dioxide fertilization effect. And the phenology is changing. The growing season is getting longer, so there's greater supply of carbon. That will drive increases, we think, in below ground carbon and soil exudation to try to increase access to nutrients. And then this will result in increased immobilization of nitrogen and nitrogen mineralization because of the increased supply of carbon, and then decreased nitrogen availability, which we think will then feed back to the uptake by plants that will increase resorption of nitrogen because of tightening up of the nitrogen cycle, which will then reduce nitrogen in leaf litter. So we see all these things going on, and we see the linkages between changes in carbon processing and nitrogen processing. But what we want to know is what is driving those changes. We're obsessed with sort of what's happening in the soil and changes in snowpack and freeze-thaw cycles and disturbance associated with that. So that's could be one line of driver. I'm skeptical about that. I think that the carbon, undoubtedly the increase in length of the growing season and increased atmospheric CO2 are probably important, but also I think recovery from acid rain, not only the decreased inputs of nitrogen that we've seen, but also increased mycorrhizal activity associated with increases in pH could be driving some of this. So this is sort of a line of research hypotheses that we have. But the other thing that I want uh, to take away from you is that we study more than nutrients. We study other elements of biogeochemistry. And they can be very important. So as I mentioned that back in the heyday of acid rain, there was a lot of sulfate and nitrate leaching. In order to maintain electroneutrality, that leaching of sulfate and nitrate drove leaching of base cations from exchange sites in soil. So what I have a plot here is the watershed flux, annual flux of different cations, calcium in blue, magnesium in red, sodium, or excuse me, magnesium in green, sodium in red, and the triangles, gray triangles of potassium as a function of strong acid anions, sulfate, nitrate, and chloride. Chloride doesn't change very much, but sulfate and nitrate have decreased over time. So early on, these were in the bad old days of high acid rain, and there was a lot of leaching of calcium and magnesium. Remember, I told you that historically the base saturation dropped about 50% from about 20 to 10. This is the process that is driving that. But as we have decreased acid deposition, that leaching has stopped, and there's less leaching from soil. The other thing that I spend a lot of time talking about and thinking about is aluminum. And so aluminum, the inorganic form of aluminum, can be toxic to fish, but it can also be toxic to plants. So one of the things we've been doing, it's been mostly me studying forms of aluminum. This is in stream water, but we've also done it for soil water. So I want you to focus on the upper panel. This is watershed 6, the reference watershed. This is watershed 1. So two forms of aluminum. Organic aluminum, which is benign in the circles, and the inorganic form of aluminum, which is in the triangles. And you can see the organic concentrations on this axis, the inorganic concentrations on that axis. So if you look here, the inorganic fraction of aluminum and the organic fraction both have dropped with increases in pH, but particularly starting around 2010 have dropped to virtually zero. So that was a big driver in terms of plant effects and microbial effects that we think, and as manifestation of the recovery from acidic deposition. The lower panel is the same thing for watershed 1. Okay, so why are we concerned about this? Well, certain tree species are very sensitive to effects of acid rain. And particularly sugar maple and red spruce. The mechanisms of this are different. Red spruce, higher elevation, it experiences a lot of cloud water chemistry, fog chemistry, which bathe the needles in those materials, which are highly acidic, and that has leached calcium from needles, which decreases this cold tolerance of the red spruce and makes it susceptible to cold tolerance. Red spruce is doing very well. Why is that? Number one, acid rain has gone way down, so that leaching is greatly diminished, but it's also got a lot warmer, so we don't have these very, very cold events that drove this winter injury. Sugar maple is a different situation. Sugar maple requires a lot of calcium, so this loss of calcium has impacted it and increased the impacts of aluminum on root function. So sugar maple is not doing well. So these are some older figures showing you the growth rate of the major tree species at Hubbard Brook. These are a little old. I'll show you some updated values in a bit. But particularly if you look at red spruce today, doing pretty well, growing pretty well. Sugar maple, doing very, very poorly. Okay, so in addition to growth, the other thing is survival. So how are the survival of tree species? So if you look at this, I've got 6 species or lump species, and the recruitment of these species is shown in green and the mortality is shown in red. So you can look at the difference between the recruitment and mortality and see how well is the survival of these individual species. So let's go through a couple of these. So this right here is balsam fir. It was doing pretty well, but recently a lot of mortality. And we think that that's because of soil moisture issues associated with the lengthening of the growing season. It does not do well under low soil moisture. We talked a little bit about red spruce, that's here. And you can see that the mortality and recruitment were pretty close, but recently the recruitment has greatly increased in red spruce. It's growing like gangbusters. It's doing very well. And it's probably because balsam fir is doing poorly and also hardwoods in that area. Plus the climate is getting better. So let's look at sugar maple. So sugar maple here is doing very poorly. You can see mortality is much, much greater than recruitment. Balsam fir, on the other hand, right here. First of all, look at the scale here. These are very, very high rates. So the recruitment has been increasing, but it's been followed by increases in uh, mortality. In fact, those have exceeded recruitment in recent years. So the forest is shifting, the major forest is shifting from sugar maple to beech, but beech is being challenged because of beech scale, as I mentioned. I also talked to you a little bit about the Wollastonite addition experiment. So this shows you, this is the data in blue is what I showed you previously, the reference watershed. The red is the above ground biomass here for watershed 1. So the trees responded very well to the Wollastonite addition, large increases in biomass. If we look at the distribution of species, I showed you this before for the reference watershed, beech doing well, sugar maple doing poorly. That's completely turned around for the Wollastonite addition. You can see sugar maple is doing very, very, very well in the treated watershed. So the last thing I want to show you is to start to talk about the interactions between these experiments and these drivers. And to do this, I've got to compare the 3 watersheds that we've talked about. The reference watershed, that's shown in black here. The whole tree harvest, it's very severe treatment shown in red, and the calcium addition experiment shown in blue. So these are stream water chemistry, pH, calcium, and silica. And so you can see that over time in the reference watershed, the pH back in the day was in the high 4s, and now it's going up to the mid 5s. So this is a manifestation of the decreases in acidic deposition, less sulfate and nitrate, and recovery in pH. In the whole tree harvest watershed in red, shortly after the cut, initially there was some acidification, but then immediately large increases in pH. And that coincided with large increases in calcium. So there was an enhance in supply of calcium at this site. And you can also see an increase in silica right here in red. So what we think is happening is in the cut watershed, there was a stimulation of weathering associated with the response to the system that drove those changes. And then I show you the watershed where we added calcium silicate, and you can see increases in calcium and increases in silica. So I'm going to skip this. We also track nitrate, and the nitrate patterns are equally interesting. So these are seasonal patterns in nitrate in stream water chemistry. So you can see the concentrations are highest in the winter months, lowest in the summer months, and then they increase in the fall. And these are for those 3 watersheds. This is data early on from the 70s to the 80s, and this is recent data. So recent data, except for watershed 1, where the nitrate concentrations have increased, the other watersheds, the nitrates concentrations have greatly decreased, which is consistent with the nitrogen oligotrophication hypothesis. So what's going on here? So I'm hoping to get some feedback from all you, but I think a big driver of this is the loss of calcium which really set the stage for the transition from sugar maple to beech. It was problematic in the reference watershed because of the inputs of acid rain, which accelerated calcium leaching. But it's particularly prominent in the whole tree harvest watershed because in addition to acid rain, those trees contained a lot of base cations, which were exported from the site. So that amplified, that harvesting amplified the calcium loss. And those shifted the conditions for mycorrhizal activities and trees that are associated with different mycorrhizal activities. So so if you look at this, these are the drivers. We added calcium here, and so we can shift the system towards a arbuscle mycorrhizal system like sugar maple, which is a more open system where we see high nitrate leaching, but it cannot deal with low calcium concentrations. Then in these sites where they're very heavily calcium impacted, that drives it towards beech, which is an ectomycorrhizal system which mines organic matter, mines mineral matter for nutrients because the nutrients are starved because of increases in calcium, decreases in nitrogen deposition. So this becomes particularly prominent in these calcium depleted sites, which takes away environment for sugar maple and makes it more conducive to beech. But the trouble with beech is beech scale. So beech scale is increasing mortality, and so rather than living 100 years, beech are only living 20 years before they're falling out. So it's changing the structure of the system. So my last slide is this very complicated diagram. And so what we think is a big driver, or my second to last slide, loss of calcium, which set the stage for this transition from maple to beech. And that, because of the mycorrhizal associations, that helps drive the nitrogen oligotrophication. But also it changes the canopy structure because beech are not living that long. So it's completely changing the dynamics of the forest system. We don't see any changes in above ground biomass, but we're seeing large increases in soil respiration. So we think a lot of the carbon from that excess carbon is being plowed below ground to try to enhance mycorrhizal activity and to obtain these nutrients that are needed to sustain this carbon input. So there's a lot going on, and we're trying to piece this all together. But you might ask the question, this is Hubbard Brook. It's a relatively small area. Is this really relevant to the northern forest? So I've got one little teaser for you. This is from the forest inventory and analysis for the Adirondacks, which is my neck of the woods in New York, as well as New England, shown in green, showing the recruitment ratio of beech to sugar maple in these FIA plants, plots. And you can see there's this huge transition away from sugar maple towards beech. So I appreciate your patience. Just a couple summaries. So I think we utilize a small watershed approach effectively at Hubbard Brook, but there are certainly limitations, and we've made long-term measurements, and these measurements have been critical to provide insight into ecosystem structure and function. We also use them for testing models and generating hypotheses. We've seen large changes in the system, certainly climate changes, temperature, precipitation, evapotranspiration, stream discharge, snowpack, and these are consistent with regional observations. Acid deposition, we knew it was having impacts when it was occurring, but even though it hasn't been prominent for a while, we can still see the legacy impacts of that and long-term effects on soil chemistry and vegetation. And we think this calcium depletion is quite important in terms of driving changes in tree species. But we're seeing this transition towards beech. Beech seem to be effective at driving ectomycorrhizal dominated weathering reactions. So maybe that process can make conditions more conducive to sugar maple going forward. But we're anxious to try to keep watching the forest and see how it responds and learning from that. So I appreciate your patience. If there are any questions, I'm happy to take them on.
All right, we have time for a few questions.
Great talk. One thing I was curious about is actually your seeming curiosity on this breaking point in respiration, like soil respiration. Could you sort of explain a little bit about what you think is going on there? Is that
Well, I can speculate. I think we have this observation. Um, we think that we know that the above ground biomass, for all intents and purposes, in the reference watershed is not changing. The species distribution are changing, but the above ground biomass, so the carbon, we think we know that there's increased carbon uptake due to a CO2 fertilization effect, as well as an increase in the growing season length. We think that carbon is being plowed underground and fueling mycorrhizal root growth, mycorrhizal activity. We are also seeing, I didn't show you any soil solution chemistry. It's a very long talk, very complicated talk. I could have made it even more so. So what I do is I look at soil solution chemistry. We're seeing some very interesting changes in carbon dynamics in the soil. We're seeing back in the day, back in the 80s, we saw large concentrations of DOC in the surface soils, and then it diminishes with depth. The patterns that we see on the surface soils, the DOC concentrations are decreasing. So they're going more towards complete mineralization. So that's part of it. But we also think that we think that the carbon that's there is being mineralized more readily. But we also think that there's more above ground carbon going below ground, which is also probably fueling this increase in restoration. So this is an active area of research. We're really interested in this. We're interested in what are the components of carbon, but there's some very interesting things going on in terms of soil development and soil carbon sequestration for sure. We have estimates of soil carbon pools, and they are declining over time. Yep. Yep.
Hi. Thank you so much for a great talk. This was so interesting. I'm really curious in the weathering signal from the clear-cut forest. Specifically, I'm interested if you could speak a little bit to how the soil solution chemistry changed and whether there is any change in base saturation following the clear cut that could be attributed to like a flushing event as opposed to a weathering event.
Yeah, so we did a lot of measurements after the clear cut. And then once we switched over to the uh, calcium silicate addition, we sort of moved away from that. We really need to go back and measure the soil chemistry. We have good measurements for several years after the clear cut, but really not into the 2000s. So we really need to do this. The other thing we need to do in the worst way is to look at the composition, the elemental composition of the vegetation. Bob Whitaker did a great job back in the 60s. We haven't done it since. We have foliar data, but we don't have bio. I'm confident that the elemental composition of the forest has changed probably quite a bit from that time, and that's a clear gap in our knowledge. So both of those, we're looking for funding to try to make those measurements. So, yeah.
Hey, Charlie, great talk. I kind of got stuck at the beginning when you said, there's a lot more rainfall and all that rainfall is coming in the summer. And that was never returned to as a major driving mechanism. So I'm curious about like what your thoughts on the major impact like that is from that. But in particular, do you guys do through fall chemistry? I mean, have you seen large changes in the transfer of material from the canopy down to the soils because of all that rainfall occurring when the canopy's out?
Yeah, Gary Lovett did through fall, and we have not done a major through fall study since that time. So that's a real hole in our information. We've got a lot of pretty good information on foliar chemistry. We just had a paper that came out on information in terms of an isotope chemistry is associated with that. It's interesting, and my sense is, so this big increase in evapotranspiration, that is an area where a lot of people at Hubbard Brick are focused on. and they think it may be due to ozone impacts. I haven't mentioned ozone. We have ozone monitoring. Ozone has dropped like a rock. It's relatively low today, so they think that may improve the function of the canopy.
Possible partly because you get more rain in the summer.
Yeah. But my simple-minded explanation is the increase in precipitation is in the summer. The growing season is getting longer. It's getting warmer. It's just going back up to the atmosphere, and part of it is undoubtedly mediated by changes in the canopy and vegetation. But I'm thinking it's just because it's rain and it's warmer. But interesting, we also have long-term soil moisture measurements, and we're seeing soil moisture deficits at the end of the season. Even though you wouldn't think this, it's getting wetter, a lot wetter, but we're seeing, and particularly at high elevation, that's why balsam fir is having problems because of soil moisture deficits. In fact, for years we've called these asbestos forests. There's a lot of people concerned about the fire, potential fire impacts because of the changes in the forest. So there's a lot we don't know, but a lot is changing very, very quickly. It's kind of mind-boggling, actually.
All right, we have time for one more quick question.
Thanks for the amazing talk, comprehensive. I was curious from all those different pieces of the puzzle, different processes and parameters that you measure, is there some that are missing in this ecosystem models trying to predict the future of this forest, or is just the complexity, all the interactions?
No, there's some things that I think are clear missing points. I was talking to Michelle about this. It's part of the reason why we're trying to get her working more at Hubbard Brook. But one thing we don't know anything about, which is kind of mind-boggling, is phosphorus. I mean, we know the phosphorus concentrations are very, very low. And given the changes in pH and all the changes that are going on in this, suggestion that maybe a lot of this mycorrhizal activity is really driven by mining for phosphorus, we know very, very little about phosphorus. And so that's a major point. And I think also just in terms of understanding the carbon dynamics and the components of carbon dynamics are something that we really need to work on in the worst way. So, yeah. Okay, we're good, Robbie?
Yep. Everyone
Okay,
give one more round of applause.
Thank you.
Okay, and students, if you want to keep this going, coffee in the dean's office conference room.
Thank you. Thank you.
Yeah, have fun in Groton.
All right. It's good to see you. I'll send you